Good afternoon, children, and welcome back to another class of biology. So, today we are going to do about Human Genome Project, HGP. Now, what is this HGP, children? It is to find out the entire set of chromosomes of an organism, which is the haploid set actually. The human genome contains about 3 into 10 to the power 10 to the power 9 base pairs. This is the haploid set. Okay. Now, Human Genome Project. It was started in 1990 and ended around 2006. Actually, was the first mega project for the sequencing of nucleotides and mapping of all the genes in human genome. Now, this project was coordinated by the US Department of Energy and the National Institute of Health. Now, what were the goals of HGP? It is to identify all the estimated genes in human DNA, which are supposed to be around 20,000 to 25,000. And then sequencing of the 3 billion chemical base pairs of the human DNA. And after sequencing, we have to store this information in computing system or databases. Okay, and then improve tools for data analysis. After storing, we have to analyze these genes, right? So we have to improve tools for data analysis. Then transfer related technologies to other sectors like industries or agriculture or medicine or forensic or like that. Okay, address the ethical, legal and social issues, which is LC, that may arise from the project. Okay, now what were the two methodologies? How was they going to find out? Were they going to sequence these genes on the chromosomes? They adopted 2 methods. Okay, first method is expressed sequence tags, which is ESTs, which is actually focused on identifying all the genes that are expressed as RNA. That is, only they were supposed to find only the genes that could be expressed as RNA. That method is called as expressed sequence tags, ESTs. Okay, and the second method was sequence annotation, which is first determining all the sequences, all the genes of the genome, then giving its place in the particular chromosome, which is called as sequence annotation. Clear, children? Okay. Now, for doing all these purpose, we need DNA from a cell or from an organism, right? Now, first you have to extract. So, we'll come to the procedure of this. So, for that, first we have to isolate DNA from a cell. And after extracting the DNA, we have to make them into random fragments, small pieces. You know, it is 2.2 meters long, our chromosomes altogether, right? And we have to cut it into many random fragments. Okay, and then these fragments are used to or introduced into a vector's body. Who is a vector, children? What is a vector? Vector is an organism or an animal which helps to carry, right? So these fragments are introduced into a vector. The commonly used vectors are bacteria and yeast, which is called as bacterial artificial chromosome or yeast artificial chromosome. Okay, now when it is introduced into the into the bacteria or yeast, it starts multiplying in the host body, right, and forms same copies, which is called as clone or cloning. Okay, right. So first, after making into fragments, it is introduced into a vector for cloning, right, and then sequencing of these fragments using automated DNA sequencers. Children, it is very difficult for human beings to sequence all these this millions of these bases together. I told it we need 3,300 books containing 1,000 pages each to write down all these sequences, right? So it is very difficult, for humanly difficult, to sequence all these. So we need an automated DNA sequencers using, what is that method? Developed by Frederick Sanger. He was a person who developed a method for sequencing of amino acid sequence in a protein, right? So this method, using Frederick Sanger method, automated DNA sequences, using sequencing the DNA using automated DNA sequences, right? Next is, then arrange the sequences based on some kind of overlapping regions. Okay, then correctly aligning each and every sequence onto the particular number of chromosome to which it belongs. The sequence belong to which of the chromosome, it is aligned to that. It is given that place, right? That is these are the procedures for how it is from isolation to aligning the position of the genes on the chromosome. Clear? Children, this is the method of doing this extraction of DNA till sequencing of the DNA or aligning of the DNA on the on the chromosomes, right? These are the methods, procedures, right? And human genome, not human genome, the genome was identified not only in human beings, and many plants and many animals genome has already been found out, right? And they are, for example, you should know all these animals, children, which are Drosophila melanogaster, rice plant, Arabidopsis, Sinor habitus, which is a free-living, non-pathogenic nematode. It's a free-living, non-pathogenic nematode. Nematode means roundworm. Okay, in these organisms also, the genome has already been gene mapping. You know what is gene mapping? Gene mapping is the arranging or aligning the genes on the chromosome in correct order, in its actual order. That is called gene mapping. So that has been done in these different organisms also. Clear? Children, what are the salient features of human genome after it was aligned, and what was found out, right? So human genome contains 3,164.7 million nucleotide bases, and total number of genes. You know what are genes? Ancestron, or which is a segment of DNA which codes for mRNA or protein. Okay, that is a gene. But there are total number of genes is about 30,000. Only 30,000 genes are present on our entire chromosome. Okay, and average gene consists of, that is the smallest of the genes consists of how many bases? It consists of 3,000 bases nearly, and the largest largest known gene in human being is about 2.4 million bases, which codes for dystrophin protein. Dystrophin. It is the longest gene or the biggest gene in human being, which contains nearly 2.4 million bases, and the smallest of the gene is the nearly 3,000. That's the smallest of all the genes, 3,000 bases. Okay, And 99.9% of every person in the world's genome is exactly same, it seems. So where is the difference? What is the difference between the different people of the world? It lies only in that 1%, 0.1 percentage, children. Not 1% also, 0.1 percentage. Okay, right. And the functions of, there are many new bases, bases on the chromosome, and many nearly the functions of over 50% of the discovered genes are unknown. They have no function at all. They are simply present. How did we get this? From where did we get this? Yeah, we got this from our parents. And where did our parents get it? From their parents. So it has been handed over from generation to generation. We can't throw it away. It is still present in our genes and they have no function at all. And we will be giving it to the next generation, of course. Right. So functions of over 50% of the discovered genes are unknown. And the chromosome 1 has most genes, which is 2968 genes is there, and Y gene of the males has the least or the fewest, which has 231 genes. Okay, and less than 2%, only 2% of the entire genome codes for proteins. And nearly more than 50% is just simply present. All the bases are simply present. There is no use at all, no function at all. Clear, children? And very large portion of human genome is made up of repeated sequences. For example, AAA or only one nitrogen base repeated millions of times, not 10 times or 20 times or 100 times, millions of times repeated. Or it can be AT or AG, whatever it is, not only, whatever, like that. Okay, so many times repeated sequences are present. Also, they have identified nearly 1.4 million locations where there are single base polymorphism. Single base. That what I told now. Either A repeated millions of times, or C like that, or T like that. Understood? Just single base mutation, which are called as SNPS, otherwise SNPS, single nucleotide polymorphism. It is called as single nucleotide polymorphism. Clear? So all these genome, the finding of the genome helps us to find in future we can able to solve many types of chromosomal disorders and things. Clear, children? Now we come to the last part of this chapter, which is the DNA fingerprinting or DNA profiling. Okay, now what is this process? It is a technique to identify the similarities and the differences of the DNA fragments of two individuals. We know everyone's genome or genes are the same on our chromosome. Only 0.1% difference is there. So to identify the differences in this 0.1% of the genes. Okay, right. And it was this process, thing was developed by Alec Jeffreys in 1985. Okay. Now, what is the use of DNA fingerprinting, children? So, it has been widely used in forensic sciences to find out culprits, to find the paternity of a child, who is the father of a child. Okay, many crimes which have been done 40-50 years back has been solved now using this DNA fingerprinting. Okay. Now, what is the use of DNA fingerprinting, children? It is used for finding out criminals. It is used widely in forensic science to find out criminals or to find the paternity of a child. Okay, so many murders which have been committed 40-50 years back has been solved now using this DNA fingerprinting, because very recently on this DNA fingerprinting has been evolved, right? So for DNA fingerprinting also, DNA of an individual is needed, right? Or if in a crime scene or from a crime scene, they may get a blood sample or a saliva or a hair or any part of the body, any body part, or any even a small piece of tissue or skin or hair is enough for getting the DNA. One cell is enough, just one cell. Okay, and they may have this police persons may have or will have wanted criminals list or suspects list. Okay, and if after finding the DNA profiling, you can match with the those suspects or the wanted list from the wanted list, and they can find out the criminals very easily, right? Now what is the basis of this? Just now from the human genome project, we noticed that human genome contains more than 50% of the our entire set, which is unwanted, which is repeated sequences. Repeated sequences are there, which are usually non-coding, which do not code for protein or which do not take part in transcription. Okay, so there are non-coding sequences. Okay, and this number of repeats is very specific from person to person. Okay, if in a child's chromosome, how did he get this the pair of chromosome? Half from the mother and half from the father's side. Okay, so if the child has the same type of repeats in one of its chromosome, it will be same from from the mother and father. Okay. So now for this DNA fingerprinting, we have to extract again the DNA from the person's cell or from the blood or whatever, from the body or from of the individual or from the crime site. We get the sample. Okay. Now it is a very long process to find the genome, entire genome of one single cell, all the 23 pairs of chromosome or 23 chromosomes, right? So we take which chromosome, what chromosome is used for this DNA fingerprinting. So only there are certain repeated portions in the genome, but when centrifugation, while doing centrifugation, these repeated portions will stand out separately as peaks, separate peaks, according to the length of the repeated sequences, according to the presence of A-T rich adenine and thymine on both the strands, right? Rich അല്ലെങ്കിൽ A and C rich segments, right? So they may stand out separately in the centrifuge, right? And this is separated. They do not use the entire set of genome. They use these peaks, which are called as satellite DNA. What is this? Satellite. They are named as mini satellite, macro satellite, like that, according to the length of the segments, according to the A and AT rich or GC, how many times, whatever it is, right? So mini satellite, macro satellite, and all those things, right? So a DNA sequence which is randomly repeated in many copy numbers is called as VNTR. VNTR. What is VNTR? Variable number tandem repeats. Okay. So these repeated sequences are also called as VNTR, and VNTR belongs to this mini satellite DNA. Okay. Now in a person, the copy number, this copy number varies in each chromosome. Each of the chromosome 1 to 23, they may have different copy number of VNTRs. You know that, right? Different copy numbers. Now, two alleles, that is paternal and maternal, of a chromosome also contain different copies, the same copies of VNTR. if it is from a child or if it's from the person or from the crime site, whatever it is, their paternal and maternal also alleles will also have the same number of VNTRs. Okay, right. Now VNTR is very specific from person to person, you know that. And the size of VNTR varies from zero point 0.1 to 20 kilobase. Okay. Children, how this VNTR has come into our genome, I told you just now that they have been caused because of mutations, repeated mutations, which is also called as polymorphism. Called as polymorphism. Right. And the polymorphism is higher in non-coding DNA or sequences because mutations in these sequences may not affect the individual's reproductive ability. Children, this is an important question. They will be asking you the steps in the process of DNA fingerprinting. You should know the steps, right? So, what is the step again? Isolation of the DNA. Then double-stranded DNA, right? Taking from any cell. Then it is digested by using, digested means splitting. Splitting into different regions by restriction endonuclease, by the enzyme called as restriction endonuclease. Then next process is separation of DNA fragments. This process divides the segment of DNA into so many fragments, many pieces, right? And the process, whether the process is proceeding properly, to understand that it is subjected to gel electrophoresis. Next aspect is gel electrophoresis. Okay, all these processes will be done in detail in the next chapter. So, I am not going to do in detail now. So, just know the steps. First is isolation of DNA. Second step is to separate the DNA fragments into small pieces by restriction endonucleases. Right. And the third step is they are subjected to gel electrophoresis. And by this process we can understand that the fragments have been made, and which is allowed to flow through a medium, gel, agar gel. Okay, and you can separate these fragments, small or big, or according to its densities you can separate this process, the segments. Okay, so what is that? By separation of DNA fragment, that is the third step. Fourth step is transferring these DNA fragments. Now the fragments, you got it, no, small and big and all those fragments, they are subjected to, or they are transferred to a nitrocellulose or nylon membrane. Okay, right, and then now they are fixed to it, and next step is to hybridize by using a radio-labeled VNTR. What is VNTR? Variable number of tandem repeats. Right. So, what is a probe, children? What is a probe now? Probe is a already known segment of DNA. We know the sequence of this DNA, sequence of bases on this DNA, which is also radio labeled, okay, which is a small segment of DNA with the known sequences. So that is used to hybridize. Hybridize means they go and join with the DNA strands. Okay, before that there is one more step, children, which is making the strands into single strand. Then only it gets hybridized. Okay, then it is subjected to autoradiography. When it's subjected to autoradiography, you can see something like our barcodes on our items we buy from hypermarket and all. No, barcodes are there. Like that type of barcode like structures you can see. Okay. Now I told you before, you have to take the DNA from the sample we get from the crime site, or the child whose paternity is to be found out, from that child's cell that chromosome is taken. Okay, from the mother's chromosome and the the supposed to be father's chromosome also we take. അതേപോലെ crime site, from crime site you get sample, and you there are suspects, from them also you take the sample, and you take only the satellite DNA from these persons, all these persons, and of chromosome 23 chromosomes are there. You can take from 1 or 2 or 3 chromosomes, you can take it, right? And you can see the match, whether that from the crime site scene you get the sample, no, from that will be matching to either of the suspect or the child's supposed to be father, whoever is the father, there will be a match between the barcodes. So it can be easily identified. So the criminals can be easily identified, and the paternity of the child also can be easily identified. These are the steps in the DNA fingerprinting at a glance, and also the applications in, that is the future use of DNA fingerprinting. All this you can have a reading. Okay, thank you children.